All right, good afternoon, everybody. Well, thanks for coming to the Resident Research Day. It's been a couple of years since we were able to have one of these. I'm thrilled that it's back again. We have a tightly packed schedule, some great research presented by the residents today. If you don't have a program, you can scan the QR code for the program for today. And the bios of the speakers are in the program that you can scan. And I'll be formally announcing Dr. Chan before her named lecture this afternoon. So without any further ado, we'd like to welcome, just a note about talks. You'll have 10 minutes. Okay, I'll give you a warning at 9 and then try to wrap it up by 10, but there's a lot of talks today. So we'll try to stay on time as best we can. All right. So the first talk today, I'd like to introduce Sarah L. Asadi, and she'll be the first speaker of the day. Sorry, I'm trying to do this with my phone. I can save paper. So Sarah's a postdoc fellow in Dr. Marcus's lab, and she'll be speaking on visual quality and accommodation with novel optical designs for myopia control. Welcome, Sarah.
So thank you for the introduction. So today I'm going to talk about visual quality and accommodation with novel design for myopia control. And this work was done in University of Rochester in collaboration with Clear Vision Company. So as we already know, myopia occurs when the image for far is focused in front of the retina. And nowadays in Europe and in the US, it's more than the 30–40% of adults are myopics. And in the Asia, more than 80% adults are myopia. And research estimate that more than half of the population around 2050 will be myopic. So it will be, it is a real concern, health concern. So the rise peak is nowadays not that much clear, but some factors like genetic, limit time spending outdoor, and demanding near touch is some of the factors that play an important role. And when we are doing a near touch, a myopic subject has like a low accommodating response that is still that the image is focused behind the retina, producing an accommodation lag, and this is triggered eye elongation. So nowadays there is different strategy to reduce myopia progression. In terms of optical treatment, we can specify spectacle like bifocal or progressive, or contact lens in terms of orthokeratology, rise ligand for overnight, or multifocal soft contact lenses. And the pharmacology style, we can specify atropine. So the most treatment is multifocal contact lens, and is based in simultaneous vision for far and near. So why multifocal design? So in multifocal design, we have two foci when we are looking for in far. The center vision is focusing the far peak on the retina, and the near is trying to make like a myopic defocus in front of the retina that inhibit signal for eye growth. But when we are doing a near touch, it can happen two situations. If the subject use the far peak and accommodate the far is the near peak is behind the retina, so we have like a myopic defocus that inhibits signal for eye growth. But if the subject use like the near peak and don't accommodate, we will trigger myopia because we will have like an image behind the retina that is contributing to eye elongation. So the design for the multifocal contact lens and how the peaks for near and far are distributed are so important to modulate and stimulate the accommodation. So in a previous study in our group, we simulate different multifocal contact lenses commercially and new designs that they are not available in the market to study the accommodation lag. In this graph, we can see the accommodation lag for different accommodation demand. If a subject doesn't have accommodation lag, we will have this line. But here we can see like an example for one subject that for no lens, we already have like some amount of accommodation lag. But when we simulate different contact lenses, we can see that some contact lenses reduce the accommodation lag, like the center distance and the spherical positive aberration, and some other design, they are putting higher the accommodation lag, like the center near and the negative spherical aberration. So the design of the multifocal contact lens is important to modulate the accommodation lag. So this, as I said, this project is in a collaboration with Clariovision. So Clariovision has the ability to write in a soft contact lens any design, changing the refractive index, and they came with an idea of 3 different contact lenses. They are not image forming, so they are 4 millimeter in the center and 4 segmented peripheral region. In the first design, there is only the focus in the 4 periphery, and the other one, they have astigmatism and coma. They are the 2 aberrations that increase in the periphery. And these patterns are suitable to simulate in a spatial light modulator. So in this study, how we can say that the design of the contact lenses and the optical quality is driven by the design. In this study, we will measure the visual acuity with a novel design that we simulate in the spatial light modulator, and also we will measure the accommodation response. And we analyze the optical visual quality, we will analyze the change in the pupil size and also the accommodation lag. So this study was done in a custom adaptive optic visual simulator here in University of Rochester. This custom adaptive optic visual simulator has like a spatial light modulator that we can simulate any pattern before manufacturing, and also has like the deformable mirror and Harman-Schack wavefront sensor that we can measure the accommodate response objectively, and also a digital micromirror device when we can simulate different stimuli. So the experiment goes, we recruit 20 subjects, then 10 myopic subject with a spherical error from -2 to -6 diopter, and 10 emmetropes with an spherical error from 0.25 and 0.75. All the subjects were having astigmatism less than 0.5 diopter, and they are healthy subjects and young. So we measured the visual acuity with a Tampere E letter in green. And before doing the visual acuity, the subject choose the best adjective focus, seeing a CMN star in green as well, and they can change the focus using a tunable lens. And we test the single vision control and also the 3 design that we simulate in the spatial light modulator. We also measure the aberration of the subject in different accommodate demands, and the accommodate demands were induced using the tunable lens. So to the stimuli, the design was simulated in green, and also we measured the aberration in infrared. So to bypass the difference between the 2 wavelengths, we subtract like a baseline, so we can get the eye response, and to analyze how the different design that we are projecting in the eye has an effect in the eye. We compute computationally each pattern with the eye response, and we crop it depending on the pupil size. So how we calculate the accommodation lag from the aberration that we measure from each subject, we compute the MTF, and the MTF is taking account the residual defocus, all the aberration of the subject, the pattern, and also the pupil size. And the accommodation lag was the difference between the peak at 0 and all the accommodation demands. So regarding the results, we can see here that the optical quality that this subject has for emmetropes and myopes was in all the cases higher than zero logmark. And we can see here that when we simulate the different contact lenses, it has like a decrease in the visual acuity, but all of them remain higher than 0 logmark. And when we calculate the visual degradation at far for each, that is the difference between the single control and each of the lenses simulated, we can see that the lens that this has less degradation is far was the design one, incomparable with the 2 others that we simulate. And for near, we try to see how how these lenses is changing and stimulate the accommodation. We can see here a myopic subject who has like a poor accommodation response, and we can see that when we simulate the different contact lenses, the accommodation response boosts and simulates in different ways depending on the design that we simulate. some design is better for some subject and for others no, but on average, we can see that mostly all the subjects prefer the design one in comparable the other one, and the standard deviation was higher in emmetropes than for myopes, and this is why, because mostly our emmetropes that when we measured, they already have like a good accommodation, and the mainly one that they benefit for booster the accommodation was were the myopic ones. When we are going for the change in the pupil size, we can see that the pupil size change decrease according to the accommodation demands for myopes and emmetropes. And when we simulate the different contact lenses, is homogenized between the two groups, the myopes and the emmetropes. And there was more slopes in myopes than in emmetropes. And in terms of accommodation lag, we can see here that the myopes has higher accommodation lag than the emmetropes, isn't accordingly with the literature. Here we can see that the slopes was higher in myopes than in emmetropes. And we can see in these two graphs how the design changed the accommodation lags, seeing that the black bar is the non-lens corresponding to the other design. We can see that all the design try to reduce the accommodation lag, and in average was the design one was the one reducing more the accommodation lag for myopes and emmetropes. And for values higher than 3 diopter, mostly all the lenses behave the same behavior. So we can conclude from this study that adaptive optic visual simulator can be reliable used to provide subjects with experience for novel design before manufacturing or trying them in eyes. And the result of the through focus visual acuity showed that design one represent the better trade-off for the 3 design that we test in terms of preserving the visual acuity and also stimulating accommodation. The pupil size change decrease according to the accommodation demands, and also in terms of accommodation lag, the design one produces the lowest lag of accommodation in terms of the 2 other design that we test in both emmetropes and myopes. And factors like subpupil size, optical design, and the native aberration of the subject should be considered by optimizing the design and bringing a balance between the visual acuity and modulation of the accommodative response. So from that, I want to thank all the people that contribute for this work, our funding, and thank you for your attention.
All right, thank you, everyone. Well, this is my last talk in residency, so I thought I'd try and make it more fun. Instead of focusing on one particular project, just sort of an overview of my research career thus far, which hasn't been that long, but hopefully there's a few decades left. And congrats to my co-residents. I'm looking forward to seeing you all tonight, many of the faculty. And if you see me in a panic, that's because I'm looking down the barrel of fellowship, and I'm actually pretty nervous about it. So what I'll do just briefly is take you on this journey that I've been in. It all started in medical school when OCTA really first hit the market. And that's when I first got interested in ophthalmology. And I started doing this research before I even really knew I wanted to be in this field. And at that time, a lot of the research really revolved around common retinal conditions we see in clinic, diabetic retinopathy, vein occlusions, and the like. And then when I came to the University of Rochester, in collaboration with Dr. Ramchandran, and Dr. Steve Silverstein, who's one of the psychologists here, we took a look at schizophrenia, which is this kind of like off-color research topic, I think, for retina specialists. And then finally, I'll be going to the University of Iowa, which is one of the meccas of inherited retinal disease, and I'm really excited about that. And I've been talking to some faculty about some potential projects there that will also focus on OCTA. So we'll start here talking about some of my experience in medical school. When OCTA first hit the market, the image acquisition was a lot slower. And in that it took patients a lot longer, more time to be able to fixate and to actually acquire the images. What we had most of the time was basically these 3x3 imaging windows that focused on the fovea, which was okay because that's where most of the action was. But, and you can see here, you have a healthy patient on the right, a diabetic eye on the left. And the great thing about OCTA is it has this really great memory. So you can see this irregular foveolar vascular zone. You can see very discrete areas of flow void or ischemia. And even if, month to month, the patient's clinical exam looks different, better or worse, maybe there's some hemorrhages that are resolving and went from looking severe to looking mild, the OCTA will have a great memory. So it's a really great way to follow patients over time. And of course, now about a decade later, much higher acquisition speed, much better depth of penetration with swept source devices now. You can get wide field images. You can see the periphery much more easily, large areas of ischemia. You can better characterize neovascularization, which here is pretty obvious, but you can see it here, some inferior NVD. And, early in the textbooks with OCTA, one of the main disadvantages that was listed was that it can't detect smaller microvascular anomalies like microaneurysms. And while it's true, it's less sensitive than FA, you actually can catch quite a few, and it really depends on which stage of the life cycle the microaneurysm is in. If it has some sort of flow in it still, some turbulence, OCTA will pick it up because it's motion. If it's sclerosed or clotted over, then it won't pick up, and you'll only see it via staining on FA. So this was one of the first projects that I worked on, and it used OCTA, but it didn't really focus on microvascular at all. What's that?
Would you like a little bit darker?
Oh, sure. So instead of focusing on the microvasculature, we focused on basically this extravascular signal, these little like splotchy red-green areas that you can see in the fovea here. And what that correlated with was these basically hyperreflective cystoid areas that were intraretinal. And you can see here, if you superimpose flow signal over it, thank you, you can see that there's one cystoid space that is a little bit more transidative and one cystoid space that's more exudative. And the OCTA was actually picking up the Brownian motion of larger lipoproteins and other particles that were in that fluid. So if you followed it over time, as that fluid coalesced, it started to basically form these little deposits that could be appreciated as hard exudate on fundus exam. And this could be seen in all sorts of exudative maculopathies that we characterized. Next, and I'm pretty sure every institute in the country was doing this, but we started using OCTA metrics to better quantify changes in some of these more common diseases, in particular diabetic retinopathy. So you can see here, you take this qualitative image that's kind of on the spectrum of grayscale. You can binarize it and get vascular density. You can also do it with skeletonization. So basically that would account for the larger vessels that could confound these calculations. You can see here, going from healthy all the way to PDR, you can see basically an increasing degree of ischemia that you can see in this 3x3 window. A little harder to appreciate in larger windows, but it was something that was easily quantified, made for some cool publications. You can also see it in other conditions like radiation retinopathy. If you compare treated eyes to fellow eyes, you can see obvious areas of ischemia, sort of this rattiness in the retina. I think I presented this before, but when you take a dosimetry map, you can actually see better on the skeletonized images that there was a decrease in vascular density in areas of higher radiation dose. So that was everything that happened in medical school. And again, I've talked about this already, so I won't go into it too much, but we started looking at schizophrenia. And when I went on interviews, this is like all people wanted to talk about, is why I was looking at this and what I was doing. And I think the big idea or thing to remember for neurodegenerative disease is like Parkinson's, Alzheimer's, and even schizophrenia, is it's really a marriage between the nervous system and the microvasculature. So instead of using MRIs and MRAs, which are expensive and time consuming and don't give you much granular detail, you can actually use the retina and OCTA to see if there's any early microvascular changes. And indeed, in patients with schizophrenia, there was changes when you look at the cohort as a whole. It was really hard to see in individuals, but pretty reliably, when you control for age, diabetes, a whole slew of factors, the patients with schizophrenia tended to have a decreased vascular density. And this isn't a particularly good example of this, but the foveolae vascular zone also had some pretty notable irregularity compared to healthy controls. And I think the clinical utility of this is still to be determined. And it's very, at this point, it's very much in its infancy, but the ultimate goal for a lot of these neurodegenerative disorders is to at least have OCTA as some sort of adjunct to help identify biomarkers or to monitor progression of some of these conditions, including schizophrenia. And so finally, I won't go into too much detail here because I haven't done it yet. But in going to Iowa, I've been speaking to some of the faculty. A lot of them are very invested in imaging, in particular OCTA. And Ed Stone and some of his colleagues and everyone there has done this sort of mind-boggling and remarkable job of characterizing and organizing this large list of inherited retinal conditions. And on top of that, they also have this giant database. I think they get swept source OCTA on most of their patients they see in clinic. So it's this rich amount of data that I'm looking forward to diving into, and I think it'll be exciting. The condition I'll focus on primarily is choroideremia, which is an X-linked condition characterized primarily by outer chorioretinal atrophy. You can see here the yellow white area in the periphery is atrophy, and that sort of orange splotch centrally is preserved retina. So they can have pretty preserved vision for a long period. So that's sort of the journey I've been on thus far and what I'm looking forward to in the future. And just a last point I'll make is that I thought the early 2020s when I started residency were going to be defined really by the pandemic, but now we're sort of entering this AI revolution, which I think is going to really transform how we use OCTA in research. I think it's going to kind of revamp a lot of the projects that have already been done and will also help us to identify some interesting pathologic features in clinic that are hard to see fundoscopically when you have a limited amount of time. So perhaps with marriage of OCTA and AI, we can really get to the bottom of some of these remaining questions that we have. And so this has just been a sampling of everything I've been through without too much depth. It was very superficial. But I just wanted to say thank you to all the faculty and colleagues here. And I really appreciate all the time you've taken to listen to me present over the past 3 years. So thank you.
All right, so next up is another one of our graduating residents, Dr. Ewing Zhao, and she will be presenting ophthalmic findings in Alström syndrome. And she's off to join a private practice in the Maryland, DC area, I believe, in the next couple of months.
Thank you. Hello, everyone. It is my pleasure to tell you a little bit about what we find about the ophthalmic findings in Alström syndrome. So Alström syndrome is a very rare autosomal recessive disorder and affects not only the vision, but also different parts of the body, including kidney, obesity, cardiomyopathy, diabetes, and hearing loss. And the prevalence of the Alström syndrome is very, very rare. It's less than 1 in 1 million. And the causative gene is Alström gene 1. But because it was a very, very rare disease, and patients has reduced life expectancy because of the multi-organ failure, and the spectrum of the clinical manifestations is often unclear. Although prior reports have shown there were, reports showing the clinical manifestations of Alström syndrome, but it was usually like smaller sample size, and often they were not consistent. And a lot of patients with Alström syndrome were previously often misdiagnosed with the common misdiagnosis includes achromatopsia, cone dystrophy, cone rod dystrophy, spasmus mutant, and even amblyopia. So our question is, what are the common ocular findings of Alström syndrome? So because it's a very rare disease, so there is biannual multidisciplinary Alström syndrome clinics at Greater Baltimore Medical Center. So from 2015 to 2023, and we gathered these data and popularized them. And those meetings were organized by Alström Syndrome International, which is a non-for-profit that benefits the patient's education and advocate for them. So during those years from 2015 to 2023, we had 47 patients, 36 children, 10 adults, and with a median age of 8, ranging from 15 months to 42 years. And their visual acuity had overall, it is definitely a progressive disease, but there is a wide variety there from 20/20 to no light perception. The percentage of patients that with light perception or worse was significantly less when they were younger, less than 8-year-old. And the youngest kid that was legally blind was 2-year-old. And the oldest patient that has useful vision was 7-year-old. So there is a great variety there. And when we look at common ophthalmic findings in kids with Armstrong syndromes, and those are the common findings, and nystagmus has been listed as the #1 findings of patients with Armstrong syndrome in both pediatric patients and adult patients. And it's very high percentage of patients that also has photophobia with the nystagmus. And not only those are very common findings, but also those are commonly the very first presenting symptoms when they were a kid. And other common findings in the retina includes vascular attenuation, and which majority of the patients with Armstrong syndrome has that. And the second most common retina findings is the internal limiting membrane reflex IOM changes in the kids. And about less than 0.5%, just a little bit, 50% of the kids have optic nerve pallor and RPE mottling. And very rare that the kids will have the classic retinal pigment counts that you might see in some other classic retinal dystrophy. So when we look at the adults, and those adults that, in addition to those common findings that were found from the kids, and they also have all those other cardinal features like vascular attenuation, IOM changes, optic nerve pallor, RPE mottling, and retinal pigment clones. So the knowledge of these ocular findings, we think is important to promptly recognize Armstrong syndrome. And hopefully you could appreciate that by comparing the adults and the kids, and the ocular phenotype of Armstrong syndrome is largely dependent on age. And this suggests that low vision interventions and potential gene-based therapies should target children with disorder. Special thanks to all the, it's a team effort, and special thanks to Dr. Klein's group from Greater Baltimore Medical Center and the low vision therapy specialist, Dr. Sunnis, also from the Greater Baltimore Medical Center. And special thanks to the Mr. and Mrs. Marshalls from the Armstrong Syndrome International. So, and this is, so Armstrong Syndrome International, their symbol is a drop of water. So they told us that because they told every patient the same, they said drop by drop the lake is formed, because what they are doing, even little things can eventually become a big thing. So thank you very much.
Alrighty, well, thank you. And so I'll be discussing a review that I worked on with Dr. Mysore about immediate sequential bilateral cataract surgery. So as a background here, traditionally we think of cataract surgery as one eye at a time on a single OR day, with the other eye coming about weeks to months later, or delayed sequential bilateral cataract surgery. It's been the standard of care, and for good reason. As a profession, we've been hesitant to explore the alternative, which is bilateral sequential cataract surgery here. It's not just because it looks a lot less cool than these bilateral shields look a lot less cool than the leader of shield here. It's largely because we have real concerns about the risks of and safety involved with bilateral cataract surgery. But as modern surgery has evolved, it's become a little bit less simple. And even here at this institution, we could think of some bilateral cases, or cases when we think that a bilateral sequential surgery would have been useful for a patient. And in those situations is really why Dr. Mysore really wanted to explore this project, to bring a good review to our legal team, as well as start the process of creating a protocol here at Flaum. So the bottom line here is that all of this is outlining why the American Academy has a preferred practice guideline statement that says that with the proper protocol, patient selection, and consent process, ISBCS is safe and evidence-based. The immediate sequential surgery is pretty rare in the US. It's few and far between, but there has been some adoption for many years in Europe. Specifically, Finland was one of the first ones here with widespread adoption in 2009. They had, 1996, actually, I'm sorry, even before that. They've had widespread adoption of immediate sequential surgery. And then in Dr. Mysore's home territory here, Canada, they are really big supporters as well. From 1996 to 2009, there is some studies published that showed 11,000 cases through 6 of the big surgical centers there in Canada. And then even in 2019 to 2020, one of those centers formed half of their cataract surgeries in immediate sequential fashion. Some of those being resident cases and resident teaching cases, which I think is really, really amazing stuff they're doing there. And then here in the US, a lot of us will probably think of, California and the Kaiser Permanente studies really performed during 2013 to 2015, publishing their review of about 3,500 patients, and usually, obviously double eyes for that. So in terms of the theoretical risks of bilateral surgery, we think of severe bilateral complications, one of those being endophthalmitis, toxic anterior segment syndrome, another big one to think about. Other things we think about is the lack of ability to adjust for that second IOL, not looking at the first eye, how that did, and just saying we're going to use the calculation for both eyes. And then just the bilateral visual recovery period, for instance, macular edema, corneal edema, those things happen after surgery, and it could be something that can happen in both eyes and really limit the patient, keep them from driving for some time. And it's really with those risks in mind that the general principles of excellence in ISBCS were created, really in direct response to those risks. There are a group of physicians, international physicians, and there's the International Society of Bilateral Cataract Surgeons. It created a protocol to help, reduce these risks. And what's amazing is that this protocol really works. When the protocol was followed, there was no reported cases of bilateral endophthalmitis or bilateral toxic anterior segment syndrome. Now there's more research to be done, more numbers to find out the actual risk, but it does work. And then a 2015 meta-analysis looked at all of those studies that used the protocol and found that there was really no increase in the complications in immediate sequential surgery compared to delayed sequential surgery. And then in regards to the IOL adjustments, the Kaiser Permanente study covered that very well, and they didn't find any difference in visual outcome between the delayed sequential and immediate sequential surgeries. Really, I think a testament to a lot of our formulas now as well. So what are some of the potential advantages? Most importantly are those realized by the patients. Decreased time off work, decreased hospital visits, decreased burden on the family that's taking care and giving those drops for like 2 months, decreased anisometropia, which is important for falls, for driving, and quicker return to binocular vision. Some patients experience great stress with surgery and obviously the risk of anesthesia. So this kind of points us to who would be the ideal candidate, someone who'd really benefit from those advantages. Those who need general anesthesia or poor anesthesia candidates, people who are pathologically anxious, have job constraints, with really high refractive errors that are going to be uncomfortable afterwards, or really at risk of safety issues if they're trying to drive with one eye that's been done and not the other. Those with family and caregiver changes as well. In terms of other potential advantages, we can look at the population-based advantages. This is theoretical now because we probably won't be planning to institute this in any way to get a population-based change, but it's an interesting thing to think about in the future as this might become more used. Even with turning over the ORs completely between both eyes, there's still a 6.5 minute save of time. And so that can be, really great for, getting our patients to the clinic. I think Dr. Chadwick's got about 3 years on his wait list right now, so it might help to get, more people through if that was where we head in the future. And then even potential future economic benefits too. Right now it's, it's reimbursing about half for the second eye, as we all probably know, but there might be a day when it reimburses more fairly, and it'd probably behoove us to be ready and have a protocol set for when that happens. So I can't go through the whole protocol, but here's some highlights that I thought were important. One being complete aseptic separation between the eyes, kind of everything except taking the patient out of the room, but redoing everything like it's a new case. That includes instrument trays and sterilization, and even looking at the sterilization trays, which batch they went through in sterilization, and making sure that there's different batches for each tray, so that if anything goes wrong in the first batch, it is not matching the second. Same with OVD and our serial numbers, making sure those serial numbers and lot numbers are different. Intracameral moxifloxacin dosing being extremely important in this case too. The ASCRS just released that statement this year about, the proper dosing of moxifloxacin. Right now, we're using that top one there, the 0.5%. And I know that we don't always measure that, 0.1 exactly, but that'll probably become crucial if we're doing bilateral eyes. And then using separate drop bottles, including patients' medications that they already have. If they are taking glaucoma medications, we should get them 2 separate drop bottles for the right and the left eye so that there's no cross-contamination. Now, not surprisingly, the consent process here is going to be extremely important. And it's where a lot of the communities that do this have focused their attention, because we are not omniscient. And we can't say that just because we haven't seen it in the studies, that it's not going to happen, that there's a severe bilateral complication. So Canada uses 1 out of a million as the quote that they give to patients for bilateral endophthalmitis, and probably what at least should be on the consent form in some way. I'm also discussing the chance of bilateral macular and corneal edema, documenting the alternatives, and that's delayed surgery. And then documenting the patient's reasons for surgery, giving the specific reason that the patient wants that immediate sequential surgery. Documenting the potential contraindications, which the protocol has a ton. Basically anything that you could think of that comes up on your cataract eval of things to be concerned about, it's a potential contraindication and something to address. And then most importantly is it should be patient driven. If there's patient uncertainty across the board, people said stop. We should really make sure the patient is sure that they want this, and sure that it's worth the risk for them specifically. So the bottom line here is that, and it's worth repeating, is that the American Academy has put this in a preferred practice guideline that with the proper protocol, patient selection, then it can be an evidence-based and good practice for our patients. And so we should be ready here at Rochester as an institute to deliver that protocol, patient selection and consent process, so that we can bring ever better care to our patients here. Thank you.
So indeed, the density of retinal circulation is unchanged in hyperglycemic mice in the first year of life. It's me, and I worked on this project with Faye, who's a graduate student in Jesse's group, and then Jesse's right over here. So our disclosures and acknowledgements. This was first presented in poster format at Arvo just this past year. So big picture view, we're looking at diabetic retinopathy, and we're comparing controls and diabetic subjects. In this case, we're using a mouse model for all the reasons people use mouse models. Very versatile, do kind of interventions perhaps that you wouldn't use in adults or in patients, accelerated time courses, etc. So in terms of background studies, many of them in IOVS, these studies have suggested that there are both structural and functional changes in diabetic mice in their first year of life, but this characterization remains incomplete. And so these include things like parasite ghosting, ghost vessels, perfusion abnormalities. But certainly there's more to be characterized in the retinal vasculature. And we focused on vascular density to further that characterization, but it's also an important metric because there is a limit or parameters to metabolic activity or integrity that are established by our vascular density. So we thought it was a pretty important thing to look at and investigate there. So what we ended up doing, again, kind of big picture here that's going on, we have our hyperglycemic mouse model, and then we had a mouse that had some of its vascular components were exhibited fluorescence across these. So we basically got our a control, a non-hyperglycemic mice, and then hyperglycemic mice, our experimental group, both of which had fluorescent components of their vasculature, which enabled us to do imaging more easily. You can see sample size of n equals 4 and then 3 fluorescent mouse. This is just a further characterization of our diabetic mouse, and I think it's actually by our next paper, our next speaker. But this is the Akita mouse. It's a popular diabetic mouse model, and really what you should be paying attention to is C here, so we can see from 4 weeks a sustained hyperglycemia in the diabetic mouses compared to the control mouse. So that's what we're working with there. Attention should be drawn to a couple things here, and my face is in the way, so let's smooth that. First is big shout out to Faye again. One of Jesse's graduate students did a ton of work on this, from which I benefited. So this slide is kind of all Faye's work. But really, the other thing that should catch your eye is really beautiful high resolution imaging of an entire retina through montaged images. These happen to be 20x magnification. So really, we're able to do with confocal microscopy, really nice high resolution imaging. For those of you coming from the clinical side like myself, who say, that doesn't quite look like a human retina, you're right. There isn't like a macula in the arcades per se. And the way we think about kind of looking at the vasculature in mice is we have the optic nerve head, which is centrally here, and then we kind of have these radial vessels coming out from that. And so when we compare vascular densities, we're looking at comparable densities from that optic nerve. My part of this project was zooming in a little bit more. We'll see that on the next slide, but again, really nice high resolution imaging at 60x mag and then submicron resolution in the Z dimension. So looking at these image cubes. So this was kind of is a summary slide of my work, and these are these image cubes or Z stacks. So these are 295 square microns, each of these, and they vary in depth, but you're looking at maybe 150 to 250 slices in the Z-axis. So Z-axis here, and this is XY, XZ here. And I ended up tracing the entire vasculature in these particular locations in diabetic mice and then in control mice at comparable distances from the optic nerve. And the colors are representative slices, if you will, from the superficial in green, intermediate in blue, and then red is the deep plexus, and you can see how that corresponds kind of most superior and intermediate and inferior, all the way over to the left. This was done in ImageJ using a plugin called SNP, and it comes up with this really cool reconstruction of the vasculature. These yellow vessels are connecting vessels and part of plexuses per se, but you can see how well the vessels stratify, and then you can see, keep getting in the way here, how that maps onto kind of the cell layers and structures that we're familiar with. So once I quantified that, we kind of came out with this, and then MATLAB was able to calculate the density in each of these plexuses and the entirety of the cube that we're looking at, and then among the connecting vessels as well. So big picture here is, again, kind of to the eye, looking at these really high-res pictures. We don't see all that much of a difference in a representative hyperglycemic and euglycemic mouse. Again, that's a little further out. And then these are kind of more of the images I was working with. And you can see again in representative cubes from hyperglycemic and euglycemic mice. Yes, there are different branching structures, but in terms of density to the eye, there isn't something where you would say, that looks remarkably different. And indeed, we have numbers to back this up. So I would focus on the top table up here, that penultimate column to the left. there you have in the hyperglycemic and euglycemic mice, we are looking at mice that are roughly controlled by age. So they're roughly the same age, and also eccentricity is that distance from the optic nerve. So comparing them at similar locations from the disc. And then this bottom table shows that is kind of what MATLAB calculated for density numbers. Technically, I mean, we can talk more about why it's millimeters and millimeters squared, which is not technically a density measurement, but that's the way to think about it. In none of the plexuses that we have here is there a significant difference between hyperglycemic and euglycemic mice, and indeed among connecting or the total vascular density. There's no significant difference between groups either. So while prior literature identifies these changes, which I discussed in the first slide, within the first year of hyperglycemia in mice, we did not find that this applied to path length specifically. No new vessel formation or dropout. This is kind of our comment and slides on the morphology. We don't notice any sort of grossly any changes to the way vessels look or new vessel formation or structure or dropout. In the last slide, we looked a little bit about vascular stratification and found that there was no preferential impact by hyperglycemia relative to others. And then just a couple of thoughts for the future. There's a lot of interesting stuff to be done in sort of relationship between vasculature and capillaries, and we can also actually see with this imaging, you can probably see all these cells. So what do kind of ratios or quantities of glial cells look like relative to capillaries and vasculature in hyperglycemic and euglycemic mice? We can use these images to start calculating that stuff. And then it does maybe offer some support for, there's often these questions in the retina about this chicken of the egg problem. Is it kind of the glial neural tissue that's maybe has a toxic effect and is causing vascular changes, or do the vascular changes precede the neural and glial changes? But given that there are some studies that show some changes in mice scotopic vision within this range of time, within this first year of life, maybe this is kind of a point that may argue for actually some neuroglial changes before the vascular changes take place. These are my references. And I do want to give a big thanks to Jesse. I basically popped into his office in this October or something like that, and I said, Jesse, I want to do research, but I have about 120 hours to do it. Do you have images for me? And we made it happen. So appreciate that. And then Faye is about 5'6", but is really standing on giant shoulders for this project. So thanks very much.
Thanks. Any questions, Phil?
Yeah.
I have a question. So how long were the mice hyperglycemic for?
It was up to 1 year. I think the earliest, our youngest mouse was like 7 weeks. And then the oldest one was like in the 50s.
Do you think that you need to carry that model out closer to the 1 year consistently to see differences? I guess it surprised me that you didn't see anything, but.
I mean, our average, if you look at our average age here, I mean, it's kind of like in the middle of that range. So 34 weeks would have been the average age of the eyes that we were looking at. But yeah, I mean, I think you certainly would see in the literature at kind of 3 months to 6 months, you could certainly start seeing changes, and our range was kind of consistent with those studies.
Yeah, thanks for the opportunity to share some of this research I've been working on with the Marcos Lab. It's been pretty interesting. This is just a pilot study that we looked at these novel collagen mimetic peptides and some applications in myopia. So myopia, it's a big problem and it's getting bigger. So these are the prevalences. It's not just a US problem. It's not just a problem in China, it's a problem all over the place. And I think Sarah stole my thunder already. And so really, it's getting bigger. It's this study by Holden, I think it was the same study you quoted. It's they're projecting 5 billion myopes by 2050. And so that's about a 50% prevalence. So I won't linger on that. You already can understand the problem. In the clinic, we think of myopia as causing bad things like, like lattice degeneration, retinal detachments, even neovascularization. And those are the big problems that we see on the clinical side. But even before those problems crop up, myopia has kind of the grassroots movement, I like to say, of these other changes that happen. There was this study that was done by Zhao and colleagues, and they did this in mice, and they looked at this this protease, MMP2, it's a metalloprotease, and they found that in not only was this upregulated in myopia, that if you cause an overexpression of this protease, that you can actually induce myopia in these in this mouse model. And so when you do that, you end up with this protease causing changes in the extracellular matrix composition. It causes decrease in type 1 collagen. So the sclera is about 90% type 1 collagen by dry weight. And that leads to decreases in this biomechanical strength and then thinning of the scleral collagenous framework, which, as you can imagine, can lead to lengthening of the globe. So is there a way to actually reverse this if you already have myopia in place? There's a lot of research that's being done looking at specifically these metalloproteases targets. But the question we wanted to answer was, okay, suppose you've already done the damage, what can you do? So we looked at these collagen mimetic peptides. They're developed by this company out in Florida, Stewart Therapeutics, and they were nice enough to give us some of their peptides. For those who are maybe a few years away from basic biochemistry, so collagen, it's generally thought of as a triple helix. You can pull it apart. You have this helical secondary structure, and the primary backbone is a repeating structure. It's a repeating function of glycine plus 2 other amino acids. I like to say X and Y. Pick your pick of what you want there. So these peptides, there's a proline, alanine, and then proline amino acid CMPs that we use, and there are just 7 repeating units here. And work that these guys had done previously, it suggested that perhaps there was some reparative effect on corneal collagen in dry eye models, looking at stabilizing the epithelium basal membrane and reducing inflammation. So kind of interesting work. And I thought, hey, can we apply this to scleral collagen and see if this has some reparative effect? And so what we did was, instead of looking directly in myopic eyes, we said, hey, can we use some surrogate for myopia, looking just at collagenase digestion of scleral collagen? So this was our experimental design. That's what we did. We found some albino rats. We didn't find them in the hospital. There was tissue that was being used by another lab, and so they were nice enough to give us their eyes. And so we had a small sample of these albino rat eyes that we dissected down to bare sclera, and then underwent this experimental procedure here. So we would look at untreated sclera and we'd image it using second harmonic generation imaging, which is kind of a known imaging technique for visualizing collagen. We would then take that sclera that we imaged, and we would take each little section, and we would digest it with a collagenase for 30 minutes, wash it, and then image it. And then we would take that same piece, undergo a treatment with these collagen mimetic peptides, either 1 or 2, and then we would image these pieces of sclera again. So once we did that, we got these images, these data stacks through the scleral thickness of the collagen organization by second harmonic generation, and we used this algorithmic method, this order coefficient that was previously developed by James Gurman in the Marcos lab, to kind of look at that organization of the collagen and kind of squish it down to one number that we could work with. So the order coefficient that we used really is it's a Fourier transform based analysis method. These are kind of the basic, I guess, modes, if you will, of that transform of how it responds to basic inputs. One way to think of it is it's a measure of like fertility of collagen fibers or straightness of collagen fibers. And so that was a useful metric that we used and applied to our images. So when we did this with the first collagen mimetic peptide, this was the proline alanine peptides. And we got these images. This is just some of the representative images from this group. You see you have untreated, and this is just like mid-depth images. Untreated, you have treated with collagenase and then treated with the CMP. And if you adjust your glasses and you squint really hard, you might say, okay, you see some curling in the untreated, it's maybe a little more straight in the collagenase, then perhaps some curling back or increased connections between the collagen fibers after CMP treatment. But tough to say. So maybe there's some subjective reordering. Really tough to say. The order coefficient was not significantly changed from this digested tissue to the treated tissue. Okay, so we tried the second collagen mimetic peptide. And again, the same kind of representative data set here. You have untreated collagenase and CMP. And here you can see perhaps a little more clearly that you do have a change in that CMP treated tissue compared to the collagenase treated tissue. And when we look at the order coefficient for this data set, what we find is that the order coefficient between untreated and collagenase statistically significantly different. Same for collagenase and CMP. Tissues 60 microns, you have this statistically significant change where you have a reversion of the treated tissue back to the native tissue. And I say it's reversion in the sense that this is morphological. So I'm not saying that the tissue is treated and then was returned to a native state, but morphologically, you have this tissue appearing more like the native state. So a very interesting result. But it does lead to a number of questions. But just briefly to summarize here, so using the second harmonic generation, we showed that there was some reorganization of the rat scleral collagen after we applied a collagenase and then the CMP2 that made it morphologically revert back to a more similar appearance to the native tissue by the second harmonic generation. And that may represent a reparative effect in this pilot study, but there's a lot of work still to be done. And so, like I said, I stress morphology because it's just what we see, but collagen is far from inert, has different binding sites, et cetera, et cetera. And so the next step that we want to look at is, okay, so we have this morphological change, but what about functionally? Is that collagen returned to native collagen in terms of strength? That's something that'd be really important for this project of myopia. And so we've been looking at introducing optical coherence tomography elastography into the lab. And that's our next step, is to look at how we can challenge this treated tissue and see if it's functionally returned to a more native state. We've also looked at better qualifying or quantifying the tissue from a texture analysis standpoint. So overall, very interesting little pilot study that I think had some positive outcome. And so we're going to move forward and to investigate this on a more broad level. But I just really like to thank the Marcos Research Group, Dr. Marcos, of course, James Gurman, who's been great, and Yakshi Zhao, who's an optics undergrad student who did a lot of work on this as well. And then just our collaborators over at Stewart Therapeutics for lending us some of their peptides. So with that, thank you very much. I appreciate your attention and letting me share my research. Any questions? So like close out here.
Excellent.
Okay, so as I was telling you all that recently our lab has published in 2020 that we can visualize leukocytes in a living mouse eye without any fluorescent labels. So today what I'm going to talk about is that actually when we track the motility of these leukocytes as they are binding or unbinding from the vascular endothelium, this provides us a new biomarker for inflammation. This is the moment of truth. So conventionally, in animal labs, when you think about studying inflammation, we enucleate the eye, we isolate the retina, and then we tag them with tag different cells or subcellular components with different fluorophores and image them under a microscope. Now, our lab does this too. So here you are seeing a DAPI stain retinal flat mount, which then when we stain with Ly6C, we can visualize neutrophils at a different wavelength. The problem with this technique is that it provides you a snapshot of inflammation. Other labs use techniques like proteomics or flow cytometry to know more about the cell composition of inflammation and other subcellular parts. But again, you lose out on the regional specificity of where the cells are coming from, where the cells are going, what they are doing inside the living retina. So what we're interested in is studying how these cells come into the retina, what do they do in the retina during inflammatory process, and how they leave the retinal tissue. So what do we need to study this in vivo without any labels? The first thing that we need is good micron level resolution. Here, what you're seeing is a conventional fundus image of an inflamed mouse retina. And when you sort of zoom down to the level where inflammatory cells might be, you really can't see much because of lack of resolution and contrast. So our lab has turned to this technology called adaptive optics, where we correct for the aberrations of the mouse eye to provide us a single unprecedented image of cellular level resolution. Now, these cells are translucent cells, and we need to render some contrast to visualize them. For this, we have adopted phase contrast technique by offsetting the detection aperture to provide sufficient contrast to visualize these cells. Now, the third thing that we need is to differentiate these cells from other cells like neurons or glia or photoreceptors. And we do that by this very unique capability of immune cells that they're highly motile. So what I'm referring to time-lapse imaging here is that we can have a very stable preparation where we can now track these immune cells as they move from seconds to minutes to hours. And with these three advances, we can actually see the leukocytes in the living mouse eye without any labels. So today I want to talk about if leukocyte motility is actually a biomarker for inflammation. And for that, we sort of go to the initiating steps of the inflammatory cascade, where once the inflammatory cue is encountered, there are adhesion molecules that are expressed both on leukocytes as well as the vascular endothelium, which enables sort of this capture of circulating leukocytes, where they now get stuck to the vascular endothelium, and they roll, crawl, arrest, and eventually they move out of the vasculature into the retinal tissue. And we can sort of see this in vivo. So here a mouse has been irritated with lipopolysaccharides to its vitreous. And what we are looking at is an inflamed mouse vein 6 hours post-injection. And you can see the cartoon cascade actually happening here, where the cells are captured at the endothelium and they're rolling and crawling along it. So this brings us to the next question. Are these cells moving at a constant speed across the vascular endothelium, or are there hotspots where they're slowing down or speeding up? And if they are slowing down or speeding up at different portions of the vessel wall, does this directly affect the next step of the inflammatory cascade, which is extravasation of leukocytes from the vasculature into the neural retina? One interesting thing that we have seen in our experiments is that there does not seem to be uniform extravasation of leukocytes from the vasculature, and we do tend to see these hotspots where the cells tend to accumulate outside the retina. So again, the question is, do these cells slow down at a certain point in the vasculature, and are those points where these cells come out? To answer this, we are going to use a mouse model of human uveitis by performing a single injection of endotoxins called lipopolysaccharides into the vitreous of the mouse eye. It serves as an irritant to the retina, and injecting LPS in the vitreous gives us a model which is both acute as well as repeatable. And I know that human uveitis describes a broad range of inflammatory targets, but when you do this intravitreal injection, our model gives us primarily a vitreal and inner retinal phenotype. Next, we take this inflamed mouse, put it under our AOSLO, and then image the veins from seconds to hours. After that, we stabilize the recordings and use ImageJ to manually track these cells as they are rolling and crawling along the vascular endothelium. And once we track these cells and plot their average velocity, we see that this is actually a very heterogeneous population. First of all, already these cells are way slower than the circulating cells by orders of magnitude. But even then, you see this sort of heterogeneity in their behavior with an average speed of about 11 microns per second. The tail end of the graph actually represents the loosely bound cells to the vasculature, which are rolling much faster. The peak of this histogram describes the more tightly bound sort of tank treading cells that have adhered to the vascular endothelium. And there are times when we see very unique behaviors like cells that are moving against the direction of flow. So today what we are asking is, are there particular portions along the vasculature where these behaviors are exhibited, or is it more stochastic? So I want you to pay attention to this cell that's marked in blue as it is rolling along the vessel wall. Now, when we track its speed as it's going along, what we see that there are parts of the vessel wall where this cell is slowing down and other parts where it's speeding up. But this is just one cell. So we tracked this particular vein for about 6 minutes and plotted the instantaneous speeds of about 70 cells here. Each cell is being represented by a different color, different colored trace. Now, what you're seeing is that not all cells are tracked across the entire vessel wall. So next, what we did was we created a sort of a gate where we considered those locations that had seen a passage of at least 5 cells. And by showing this, what I'm trying to get across is that we now have both the positional as well as the velocity information to say that there are microdomains where cells slow down and cells speed up. Here you are seeing at least 2 domains. In the center, you have the fast domain where leukocytes are losing their adhesion, and it's flanked by these 2 slower domains where there seem to be more expression of either adhesion molecules, or there is something unique going on. And this is not just one vessel. This is the top part of the same vessel, but for simplicity's sake, I'm only showing the average trace of speed as the cells move along. And again, what we are seeing here is that most of the cells start out very strongly adhered at the start of this vessel, and then they lose their adhesiveness. We also, so what I showed you before was for about 6 minutes, we tracked vessels over a period of an hour, and we see that these microdomains are remarkably consistent over the period of an hour. So here you are seeing that over 60 minutes, there across the central portion of the cell of the vessel, the cells were consistently slowing down, and then they were speeding up as they move along. So what does this tell you? This is one of the first measurements that we are seeing where cellular leukocyte motility can be used as a biomarker for inflammation. And this is really the first step. As I said, the next step would be to see if the portions where these cells slow down are actually where these cells migrate into the retina. The next question would be, what is so special about these microdomains where the cells are slowing down? Are there pericytes there, or are there microglia there? And our lab has the capability to image simultaneously both fluorescence as well as in near infrared wavelengths, different kinds of cells. So to summarize, we can see rolling leukocytes with phase contrast with adaptive optics, and we can identify these domains where they along the vessel wall where these cells are either speeding up or slowing down. And this sort of reveals an invisible gradient of greater or lesser adhesion molecule expression and can be used as a biomarker for inflammation. That's
Thank you very much. That was outstanding.
Question. So I was curious, does the morphology of the vessel and the turbulence of the flow in that vessel, you think impact the speed of your leukocytes?
question. So we have seen that the diameter of the vessels do change as these are all primarily veins, so the diameters do change to accommodate for flow. So that might be one of the reasons why this is why we are seeing these microdomains, but primarily they will stick only if there is an expression of adhesion molecules there. So if there are selectins and integrins being expressed, that's the only time when these cells are going to stick to the vessel wall. So while the diameter of flow might affect, the primary reason would be the molecule expression.
So thank you for that introduction. We'll see if I finish residency. This is a really long title, but we'll break it down slowly here. No disclosures because I'm a resident. A little background. Presbyopia, as you know, is kind of age-related loss of elasticity of the lens, causing a loss of accommodation. And so normally, when you're younger, when you're trying to look at something close by, the lens is able to become more spherical and able to converge those light rays better, focusing on the fovea, letting you able to see. As you get older, though, unfortunately, the lens becomes more rigid and unable to become more spherical, and hence you become more hyperopic when trying to read things with age. So there's certain treatments that we do, at least traditionally in clinic, as you know, is we typically say, well, get some lenses, right? Go to Walmart, buy some reading glasses, or some people use contact lenses. There's a slightly more recent trend of using a really old eye drug called pilocarpine to do a pharmacological pinhole effect to help with reading as well. And then there's some surgical options, LASIK, for example, presbyLASIK, conductive keratoplasty, and some corneal inlays as well, which is what I'll talk about here. So the one I'm going to talk about today briefly is the raindrop near vision inlay. And if you've been up to date with these things, it was approved in 2016 as
Why don't you stop just asking them to mute themselves?
can you please mute yourself, whoever you
If you're
are?
on Zoom, please mute yourself.
Or Steve might be able to do
I'm
that.
gonna mute all.
Oh,
Okay.
And then
I was like, is people laughing? I said, I didn't think I did much. It's a corneal reshaping inlaid hydrogel. And eventually I will click and it will move or nothing. Okay, yes. So, and what it is you make a little LASIK flap and you effectively put this hydrogel in and it creates a hyperprolate anterior corneal surface. And what this does, it helps converge those rays, help with presbyopia and corrects with that near vision acuity, primarily implanted in one eye, in the non-dominant eye to correct for presbyopia. But as I mentioned earlier, though, in 2018, 2 years later, there was a class 1 recall for post-operative haze. That was based on the 5-year post-approval study, which they found that 75% of the patients who had this ended up with corneal haze, which apparently is bad. So, the purpose of our study was to try to see, you know, after this post-FDA recall, you know, how you deal with this corneal haze and the induced hyperopia we'll talk about in a second. And so the methods here is a retrospective, a non-comparative study of 16 patients who had this implant put in. And then we did a comparison of the visual outcomes of these patients pre and post implantation, explantation, et cetera, which I'll demonstrate in a second here. So here's just the general demographics of these patients. And as you can see, the two things I really want to point out here is, first of all, the average age of patients, as you can imagine, are presbyopic. The average age was 54. And the duration of the inlay was in about 1.5 years or so. And all these patients generally were a little bit hyperopic when we first did this. And so the confusing part about this a little bit is kind of like the timing of all the periods. So I'm going to go over it really briefly. And so the first initial period is pre-implantation, when we first evaluated these patients to see whether or not they should get this inlay. Then afterwards, we put the inlay in. We then checked the outcomes 1 month later to see how they're doing. That said, in the middle here, there was the FDA recall. And so we evaluated those patients, and in the recall they said that there was 2 options: either we could explant it or we can monitor you closely. In our case, we explanted all of them. And so we had the visit right before we explanted them, that, 1.5 years afterwards. And then, we want to see a month after what happened. And so here's the visual acuity, and this is visual acuity during the implantation course. And as mentioned before, these patients were a little bit hyperopic, and they actually trended towards eagle vision, right? They're closer to the 2015 line. And then you can see after implantation, they became a little bit, their distant vision became a little bit worse. And that's because actually their near vision became a little bit better. And we saw that trend actually go across during the post-implantation phase all the way to the post-explantation phase. But what's more interesting is kind of the refractive change during the implantation course. So as I mentioned before, initially those patients were a little bit hyperopic, and the goal of putting these implants in was, well, to make them a little bit more myopic, right? So they can see a little bit closer with that eye. And that's exactly what we achieved. And that was what the thing was supposed to do, right? And so even 1.5 years later, that trend was still there. They were still a little bit myopic, and patients actually still liked it. But the most interesting thing is this one back here, and though it's a trend, we'll explore this a little bit more. You'll see that after we took out the inlay, you can see that they were not only returned to their hyperopia, but they actually became a little bit more hyperopic as well. And so we wanted to explore why that was, right? And so we followed these patients for a bit, and we kept checking them for changes in refraction. And the decision was, if they became more hyperopic over time, that we would decide to retreat them either with hyperopic LASIK or PRK. And then we would actually check them a month after that treatment as well. So like I said, lots of time points, hopefully I explained that okay. And so this is probably the most interesting chart. So as I mentioned, those patients were initially hyperopic, became myopic, that was the goal, became a little bit more hyperopic than they were before, after we explanted them. But we split these patients, as you can see, some patients after explantation were basically plano. They were perfect. We didn't have to do anything to them. But there was another subset of patients over here you can see. So these are the ones we didn't treat, that became hyperopic even 1 month after we explanted the inlay. And we continue to follow these patients, see just progression, checking for stabilization. And you can see by the time we decided to retreat them, a lot of them became a lot more hyperopic, even more so than the data that we showed originally. And, so of those patients, so 7 of the 16 patients had that treatment, they were able to return back to Plano afterwards. And so the question is why, right? Why did this happen? And so, as mentioned previously, 7 patients got hyperopic LASIK or PRK. So that's the distribution right there. But as mentioned before, these patients were pulled because of corneal haze. And so the thought was maybe that all the patients that became more hyperopic over time had corneal haze versus, the ones that didn't have the haze. And so it's a little bit of speculation, and we do have some of the topographic data that I'm not showing today, but the thought is maybe that this hazing is actually midstromal scarring and contraction, causing kind of a flattening effect that's causing a little bit more hyperopia. So this is something that we're going to explore over time. But what we've shown is that, of the patients who do experience this, that they are responsive to that hyperopic LASIK or PRK refractive surgery to get them back to where they were before. So with that, here are my references. And I just want to thank everyone, including Dr. McRae, for all his help with this project.
So to start with, if the slide will advance, I will give you some background on the UEC and just resident clinics in general in the US. Residency training began in 1889, so it's been around for a long time. Since its establishment, there's been a central tension between the motivation to provide excellent training for future physicians, but also wanting to provide the best quality care for all of our patients. The reason this is particularly tense is that 75% of academic health centers are located in underserved communities with patients who are underinsured or uninsured. We know this with the resident clinic that used to be here at Flaum. What ends up happening across the country, not just here, not just now, is that the average academic residency clinic patient has a longer wait time, a poor continuity of care with an attending physician, and inadequate resources considering relative lack of staff, lack of infrastructure in resident clinics. And keep in mind, these patients are the ones who are most likely to have financial, physical, and emotional barriers to optimal self-care. So this is sort of the background of what an academic resident clinic is. And all this being said, programs in all specialties across the country are working on reimagining their resident clinics in the name of health equity and justice. This has been something that's been going on over the last few years. So our resident clinic patients just in July were absorbed into faculty practices with the idea that they'd have improved continuity of care with a attending ophthalmologist. But then the question becomes, I'm a resident, there are residents here, there are previous residents, there are future residents in the room. What do we do with the residents and their education? With all of those resident clinic spots, we've created the UEC. So residents still see patients in their own little area, but now it's only urgent patients and same day visits, as the biggest issue with the traditional resident clinic is the poor continuity. So as I said, why UEC? This is the health equity issue, continuity of care issue. And then if anyone's interested in sort of the history of academic residency clinics, it's a great article in the New England Journal of Medicine. I can send at your request to your email. So now I'm coming to the objective of my project. How does the UEC impact education of PGY2 trainees in terms of patient volume, the breadth of clinical experience, and their preparedness for the PGY3 level? So as an incoming PGY2, I have a vested interest in this. I'll be spending 6 months of the year in this space. And I was hoping that this project would shed some light on its relative strengths and areas for improvement. Methods from this time period, this is July 1st of 2022 to November 30th of 2022, the first 5 months of the UEC. I was able to take out every single UEC clinic visit that was seen by a PGY2 or PGY3 resident, and a smattering of PGY4 visits as well. And then I took the primary diagnoses listed for these visits and I categorized them into several subspecialties that are listed here. I can go into more detail if anyone is curious, but general ophthalmology, I basically thought in practice, would I send this person and follow up to a specialist, or would I handle it in my own comprehensive practice? And then the other ones you can see are trauma, cornea, anterior segment, retina, glaucoma, uveitis, neuro-ophthalmology, and Other is a truly interesting category, a lot of headaches and non-ophthalmologic complaints. And then I also went about these surveys of the PGY2 and PGY3 residents. I had 100% completion. Thank you, guys. I showed up with papers to the clinic. So the surveys were also very brief, which was I think helpful for getting them all done. It was just these 3 questions, or these 3 statements rather. The volume of patients seen is appropriate for PGY3 preparedness. The breadth of pathology is appropriate for PGY3 preparedness. And a general all-encompassing PGY2 residents are prepared for PGY3. And then for those, people were asked to rate on the standard 1 to 5 scale from strongly disagree to strongly agree. Again, this is a self-assessment of the PGY2 residents and the PGY3 residents who were in that space. So now I will discuss the results. I have a pie chart here. This is my results. So these are the visit diagnoses in the UEC by subspecialty. This image is supposed to represent the breadth of pathology that's seen in the UEC. And I think people can maybe mentally compare this to a traditional comprehensive resident clinic. As you can see, general complaints still make up the largest part of this, that's 33%. The most common diagnoses in this category were dry eye, conjunctivitis, styes, and then some patients who actually had no physical exam finding, but while they were there, just had a comprehensive eye exam because they were reporting blurry vision. And then the next runner up here is other. I won't go into that because I think that that's mainly poor documentation or just completely non-ophthalmologic complaints. Neuro-ophthalmology made up 11% of the visits, which is much higher than you would expect in a standard comprehensive or a standard traditional resident clinic. And then as you can see, some of the other big differences are that only 2% of patients had a primary visit diagnosis of glaucoma. This is very different from a traditional comprehensive resident clinic. And we see actually quite a bit more uveitis than we would in a traditional resident clinic. That is 6%, or that is 8.3 uveitis cases per resident in a 5-month period. So that's quite a bit more uveitis than we would expect. Volume, this was the other piece of it, aside from breadth, which was discussed in the previous slide. So during the study period, the average number of patients per resident was 131 plus or minus 31 patients. So this comes out to just under 300 patients a year. As I was preparing this presentation, it occurred to me that there is an inpatient resident and a UEC resident at all times. So they are disproportionately represented in this 131 number. But if I took it out over the course of a year, I think it would average out. So as you can see, the volume is a little bit different than what you'd expect in also a traditional resident clinic. Survey results, again, thank you guys for the 100% completion rate. Survey results indicated that most trainees were neutral regarding the total volume as it pertains to PGY3 preparedness. But most people agreed that the breadth of pathology seen in the UEC was appropriate for preparedness for PGY3. And then I also included some open space for the residents on their surveys to include their thoughts. I really appreciate the effort that was put into them. Some people even went on to the back of the page. So thank you guys. Across the board, most people said that the breadth of pathology was the biggest strength in the UEC, and that there was increased interaction as a PGY2 resident with all subspecialties of ophthalmology. Because we offer a same-day appointment, we are obviously more biased towards seeing ophthalmologic emergencies or things that acutely cause pain or vision loss. And as a result, we frequently get to talk to our uveitis attendings, or we ask our retina fellow or attending for assistance with urgent intervention, or we staff with neuro-ophthalmology, and all of this is considered to be an educational benefit over a traditional comprehensive resident clinic. Another strength was from the call perspective. PGY2 stated that because there is a more urgent emphasis on the UEC, they're far more comfortable with call, as by the time call had picked up, they had already seen a lot of the common urgent and acute eye complaints in the clinic setting with all the supervision around them with their upper levels and attendings. However, no system is without areas for improvement. I've included a few here that were discussed in the surveys. The most reported area for improvement is probably volume. That was mentioned in most of the surveys. While a busy day in the UEC, Friday before a long weekend, any Friday can quickly overwhelm our capacity. There are days of the week where the volume is still very low, and this is something that we're actively working on. Something that goes hand in hand with volume is definitely triaging. So when you have a low volume, you worry that your triage is missing people. And when you have a high volume, you worry that your triage is letting every patient be an urgent patient. I'm sure that people have heard about the triaging in the UEC enough, so I won't belabor the point, but the quantity of patients that are seen for headaches or chronic dry eye is still high because they report acute onset eye pain. And then ultimately, a morning comprised of these patients is not as educational as a morning of bread and butter comprehensive eye exams. Lastly, continuity. Being able to see the same patient for years in continuity is unfortunately actually not realistic in any resident clinic because we rotate through different rotations. So this is something that is, I think it's just not going to be possible in either this clinic or another clinic. So any way to go about this would be, I'd be interested in hearing about. So for the future, wrapping up here, increased volume. We're about to start advertising in the community. So this is about to change the makeup of the UEC quite a bit. PCP offices and urgent cares are going to find out we exist and send their eye patients to us directly. With this comes the question of triaging, what are we going to do about the influx of calls, and what we will do is adapt. So I'd like to add here that I've actually obtained IRB approval to pull data on every single patient seen in the UEC. So every single patient that has been seen in the urgent eye care, I have their demographic details and visit information like their diagnosis, billing level, whether or not an interpreter was used, what their insurance status is. So I have a couple plans for the data. If anyone has a specific interest in the UEC or has some research interest that they would like to use this data set for, please let me know. And then special thanks to Drs. Wozniak, Plotnick, and Wendy for helping me kind of merge my interest in my own education with my interest in health equity in this project.
So just to dive right in, this research was supported by various different grants, both in the United States and with the Spanish government, given Dr. Marcos' affiliation with this project. So the main purpose of this project was really to determine the structural changes of the crystalline lens that we see with age, and then also refraction as well. And the main methodology that we used in this study was looking at anterior segment OCT images. And this was a population of cataract surgery patients. So it's important because when we think about the preoperative things that we want to consider, imaging of the crystalline lens is very important in terms of our preoperative calculations for the IOLs, effective lens positioning, and as I'll touch on later, various different future applications with this. So just to give a little background in terms of how we actually image the crystalline lens, there are a lot of different challenges with this type of imaging. Oh. There's a lot of different challenges with this kind of imaging. And part of it is because of the anatomical limitations that we have with our current optical techniques. So if you look at this image here, this is just an OCT image of the anterior segment. And if you wanted to access the lens, you're really only able to see the portion that's visible in the pupil. The iris and the ciliary body block a lot of the imaging that we can get there. You're able to get a full 3D image of the optical of the lens using more non-optical techniques, so like MRI or UBM, but those are limited by acquisition time with MRI. You don't have great resolution with the MRI either. And when you're doing a UBM, it's really based on the skill of the examiner. And then when we think about the actual images that we obtain, like say with OCT, there are a lot of optical distortions that you have to consider in terms of quantitatively analyzing the lens. So this study is really based off of a lot of work that's been done by Susanna Marcos' group and their various colleagues over the course of the last 10 years pretty much. So I'll just give a general overview of how we actually got to getting a 3D model of the crystalline lens. So it all started by first imaging about 133 actual crystalline lenses in vivo. So they did that by isolating the lens from human donors and then using an OCT to image both anterior up orientation, so the anterior portion upwards, and then they would flip the lens around and get a posterior up orientation as well, just to be able to get a full idea of what the crystalline lens looks like in vivo. And they did that across various different ages, going all the way from newborns to about 71 years of age. And from those raw images, they were actually able to take those images and do surface segmentation. So doing like some edge detection and getting a sense of what the outside structure of the crystalline lens looks like. And then using that data, they were able to do some more corrections for certain optical distortions and fan distortions that are induced by that specific imaging technique. And then from there, they essentially took those anterior up positions of the crystalline lens and merged them with the posterior up to get a full crystalline lens shape in 3 dimensions. So from there, the next step was really to form an idea of what the average lens may look like. So using those 133 crystalline lenses, they were able to get a general sense of what you can extrapolate the lens to be. There's a lot of math involved in this and a lot of like weird shapes and figures that I'm not going to get into. But essentially, they were able to come up with an average crystalline lens shape. And then looking at this side of the figure, they were able to use the anterior and posterior portions that were within the first like 4 millimeters or so, kind of simulating what you would see with a pupil to also extrapolate the lens. And they also generated different types of lenses across different age groups. And overall, really the main thing with this is that they were able to accurately determine what the 3D crystalline lens shape was using very limited parameters that we can get through the OCT, which is just those kind of central 4 to 5 millimeters or so of space. And in the previous work that they did, they used these models that they generated to train over time by imaging in vivo and then comparing it to actual crystalline lenses that they had taken the previous images from. And over time, they were able to get a very accurate model that we could use to be able to get a 3D geometric shape of the crystalline lens with the OCT images. So bringing that all back to the project that I was working on mostly, we were looking at 41 eyes and 21 subjects who were about to go undergo cataract surgery. And we just took preoperative normal OCT image scans with the Iowa Master 700. And we also collected their preoperative manifest refraction as well. And using that, we were able to use their preoperative OCT images, do that same surface segmentation, and then generate a 3D model or 3D construction of the crystalline lens in those patients. And from that, we were able to get various different OCT-based features that we could quantify their actual crystalline lens. So we could get their crystalline lens thickness from there, their axial length, which was also reported from the OCT. But more importantly, we were able to estimate the radius of curvature of the anterior lens surface, the radius of curvature of the posterior lens surface, the total lens volume, and then also more importantly, the lens equatorial diameter, which you can't really get with the standard OCT image. You really have to extrapolate. And then as well as we also collected their pupil diameter as well. And so to dive into some of the results, one thing that we did notice is with increasing age, the lens thickens as well with age. And then we did find that the posterior curvature of the lens actually steepens with age as well. So the radius of the posterior curvature of the lens decreases, which represents steepening. And then as expected, we found a correlation with pupil diameter and age. Pupil diameter starts to decrease with age as expected. And then when we look at refraction, with increasing myopia and increasing axial length, kind of a surrogate of both of those, we found that the posterior lens surface actually flattened. We didn't really see many changes in terms of the anterior surface in terms of both age and refraction, but most of the changes that we were observing were in the posterior curvature. And then as well, if we look here, the axial length also increased. As the axial length increased, the crystalline lens volume and the equatorial diameter were also observed to increase as well. So really, the most important thing about this is really where we can take this 3D model and apply it to different functions. So one of the big things that we're going to be doing moving forward is applying this 3D crystalline lens model to help predict the effective lens position so that you can better estimate where your lens is, your IOL is going to be after cataract surgery. And with that also comes better IOL power formula calculations. And then you can apply that same model to other structures such as like sizing for ICLs or phakic IOLs, being able to get a good sense of like the 3D crystalline lens structure and where that ICL may be positioned to be able to figure out what diameter or what size would work for individual patients. And then we could think of other possible future applications such as assessing accommodation, being able to get a sense of how the crystalline lens actually changes with accommodation, and then also being able to scan patients and see, well, is this patient losing accommodation for a certain reason because of their the structure of their crystalline lens changing. And then similarly, also if we think about myopia, myopia development and treatment as well, we don't really know much about how the lens plays a role in that. We think more about like the cornea, for instance, but in terms of actual development of myopia, we can be able to better estimate how the 3D crystalline lens changes shape with that, and then also to better evaluate treatments as well to see if that's affecting the lens shape as well. So with that, I want to thank all of the co-authors that were part of this project, in particular, Dr. Marcos, Dr. Martinez Enrique, Dr. Zhao, Dr. Chelik, and Dr. McRae, and Dr. Sitin, and all those others that were involved in the previous work that led up to creating that model. And these are my references.
So this presentation will be based on a review paper that Dr. Wozniak and I wrote about three-dimensional heads-up cataract surgery for the journal Advances in Ophthalmology and Optometry. So essentially, what is heads-up, 3D heads-up surgery? It basically involves a surgical field viewed on a 3D display screen as opposed to through a traditional binocular viewing microscope. And, you know, it's called heads up because it's thought that you're able to have more kind of positional freedom as you're not constricted to the viewing scope. And so you can see this diagram on the right. So the 3D heads up system, it consists of 1 or 2 cameras, a central processing unit, a display monitor, as shown here, and 3D polarized glasses. And the camera is attached to the operating microscope and it captures the individual field of view from each ocular. Images are then transmitted to the central processing unit, which organizes and recreates the entire operating field on the display monitor. The observers in the room wear 3D polarized glasses, and the right eyeglasses lens, this is, you can have different ways of polarizing. You could use like horizontal or vertical polarization. Circular polarization is something that's being used more minimally, but for this example, the right eyeglasses lens is vertically polarized. The left eyeglasses lens is horizontally polarized. And then you can see, so odd numbered rows are on the display screen are vertically polarized, even number rows horizontally polarized. And you can kind of imagine images from vertically polarized rows going and eventually being kind of going through the black arrows here. And then those vertically polarized images are seen by the right eye. And in contrast, you know, you have these even number rows with horizontally polarized, that are horizontally polarized, kind of going through this gray path and being observed by the left eye. And so as a result, when a viewer is looking at the display screen, the observer, they achieve stereopsis because there's slightly different images from each eye. And that's kind of the difference between this type of display and just looking at a regular screen. So in terms of advantages,
one of the biggest ones is ergonomics. So heads-up surgery offers a solution to an extremely common problem in the field of ophthalmology, musculoskeletal pain. And the prevalence of neck and back pain in eye doctors in studies has ranged from 51.8% to 81%. And in heads-up surgery, surgeons can adopt a more comfortable and neutral position. There's also the benefit of educational value. A 3D heads-up display allows for an enhanced view of the surgical field by all surgical team members, including surgical technicians, scrub nurses, medical students, and I've even seen anesthesiologists enjoy the view. This can promote improved teamwork and coordination, as well as an improvement in terms of the trainee educational experience. Visualization is another advantage. So heads-up systems are thought to be associated with better visualization of the surgical field due to superior depth perception, and improved quality. Another advantage is that there is less retinal phototoxicity because there's lower light illumination levels required for heads-up display systems than traditional microscopes. But despite having lower light illumination levels, they produce brighter images. And this can lead to a difference in macular light exposure, and that may contribute to early visual acuity recovery following cataract surgery. And then kind of the last advantage that I'm going to talk about is the ability for 3D heads-up display systems to display data or other ophthalmic imaging either next to or laid on top of the surgical field on the display monitor. And moving on to the disadvantages. The first is the learning curve. So ophthalmologists may not want to consider learning a new way to operate, given that they spent so much time and effort into mastering surgery under a conventional microscope. Crosstalk. So crosstalk, it's a complicated phenomenon that basically leads to blurring of images. And there's an importance of calculating the proper distance between an observer and the display screen and figuring out the perfect angle by which they should be viewing the screen. And if they're not within certain confines, you can have blurring of the image, which obviously isn't acceptable. There's a high cost associated with this. There are a few different heads-up visualization systems, but they really range from about $50,000 to $100,000. And then this one, I think, is another thing that has been talked about a lot in the field, image latency. So there's a 50 millisecond to 80 millisecond image latency, and those are in the two kind of most common 3D heads-up visualization systems. And surgeons have been polled a lot actually about whether they found this noticeable, and most have said they don't, but the time delay kind of changes depending on what type of surgery you're doing. So in anterior segment surgery, there's more rapid surgical steps compared to posterior segment surgery, and as a result, you may be more likely to kind of pick up on that image latency. So in terms of future directions, heads-up visualization systems could incorporate artificial intelligence algorithms with intraoperative data to make cataract surgery safer and more effective. And those are my references.
So it's my pleasure to welcome our guest speaker today, Matilda Chan. Matilda was I started in 2003 here, and I think she started in 2003. We crossed paths also at the Keck School of Medicine. When I was a resident, she was a medical student. And so she was in the first graduating class when I was attending here. And she's gone on to do great work. So it's a particular pleasure that I introduce her today as our guest speaker. Many of you in the audience know her from her days in residency here. And she is one of the first residents who came to Rochester when Steve Eldon established our program. It's been doing us proud ever since. She received her undergraduate degree from UC Berkeley, went on to complete her medical training and doctoral degree in biochemistry at USC Keck School of Medicine. And she came to Rochester for ophthalmology residency under the directorship of Mina Chung. And she was a delight to work with. And as she developed her clinical skills and research interests, graduating in the class of 2006. A couple years behind you, Fred. With a strong interest in cornea, she was accepted into cornea uveitis fellowship at UC San Francisco's Proctor Foundation. Upon completing her fellowship, she joined UCSF faculty as a clinical instructor of ophthalmology and began her ascension up the academic ladder while completing a postdoctoral fellowship in anatomy under Zena Werb. In 2021, she was appointed professor of ophthalmology at UCSF. Matilda is a triple threat embodiment of a clinician, researcher, and teacher. At Proctor, she sees patients with complex corneal disease. She also sees patients at San Francisco General and is an attending ophthalmologist for the UCSF ophthalmology and optometry programs. She has also become a prolific and highly respected cornea scientist studying the molecular mechanisms important in the development of corneal opacification. Here, her primary focuses are on corneal fibrosis in response to injury and Fuchs. She's been recognized through numerous grants from institutions such as the RPB, That Men May See, and the National Eye Institute, and she's currently funded by her second R01. These efforts have resulted in more than 40 peer-reviewed publication and appointment as chair of an NIH study section that is related to aging and development. Matilda has truly given back to ophthalmology through teaching. Since beginning her career at UCSF, she's consistently been a lecturer in the medical school as well as a mentor in the clinic. She's also supervised or mentored more than 40 undergraduates, medical students, doctoral candidates, residents, fellows, postdocs, and junior faculty, and actively presents at national and international clinical and research meetings. Really excited to have you back here, Matilda. Thank you for taking the long journey out, and she'll be presenting Bench and Bedside by Adventures After Residency. Welcome.
That's a very generous introduction. It's been so nice to be back. I can't believe for the longest time I thought I'd only been gone about 10 years. And then when I actually did the calculation, I realized it was actually 17. And it's been so nice seeing old friends. It's also been super nice meeting new friends today, and so many familiar faces. And the research that's been presented today has been spectacular. I'm really impressed. All this stuff, probably some of it happened during COVID, which makes it an extra challenge, which makes it extra impressive. And, you know, Rochester's always been known for their imaging and optics, and it was very clear today that it's still amazing here for those technologies. So I will just start off by saying I don't have any financial disclosures related to this presentation. So this is where my journey began. So I think Dr. DiLoretto gave a little bit of an introduction to this, but I don't know how many people in this audience recognize this building, but this is LA County, USC. So this is where I did my medical school. And when I was a third year medical school, I was at the tail end of my third year medical school, and I had decided I wanted to have research be a major part of my career. So I had decided on doing a residency in pathology, and I happened to do a 1-week rotation in ophthalmology. And that was the very first time I met Dr. DiLoretto. He was a third year resident. I was a third year medical student. So this was the tail end of my third year of medical school, and I wanted to make this. I didn't know anything about ophthalmology. Loved this 1-week rotation, so I had to scramble at the last minute for a sub internship, which very generously Dr. Felden took me under his wings and just and became my mentor for my sub-I between my third and fourth year medical school. And during that sub-I, that's when I met Mina. And I remember even as a medical student, Dr. Felden took me into the OR with Mina. And I just remember the two of them just sitting side by side, operating together. And it was clear to me that that was the direction that I wanted to go in. So that takes us now to residency. So I was very fortunate during residency to have amazing mentors. And at that time it was Dr. Geringer he took over the educational aspect of our residency program. Dr. Duche, of course, was there from day one and is still there. It's just been amazing. Like I'll see patients to this day and I'll still remember, especially when I need to go and refract a patient, I'll think this vision doesn't quite match what I'm finding clinically, and I'll think, I know Dr. Duche taught me how to refract. This is what I have to do. And oftentimes it was the right thing to do. Dr. Kleiman and Dr. Bogani, who was incredibly patient with me in the operating room. I was super lucky to have a really, really nice group of co-residents during my time, some of which have actually stayed in the area. And you guys are very fortunate to have them still here as colleagues. I was also very lucky during my residency to have become, to have made friends. So Crystal Huxlin was my cornea role model for corneal wound healing. She used to have these cats that walked around in her lab with things sticking out of their head. Lynn Yan, who I worked with way back in residency, and actually this last cycle for the for the NIH grant submissions, we actually submitted, even all these years later, we actually submitted a grant together as co-PIs for an R21 application. So we got scored, so hopefully that'll let us work together in the future. And then very briefly, I worked with David Calkins during my first year of residency, and we were actually able to publish that paper, and that paper actually helped me go on with getting more grant applications. And to this day, he still remains a really, really nice supporter and mentor for my research activities. And finally, this was the group that convinced me to go into cornea. It was a great group. Dr. Ching, who I'm so lucky that it turned out that he happened to be in town right when I was visiting. So nice to see him again. And Dr. Aquavella, of course, Dr. Plotnick, and Dr. McCrae. So when I was a second year resident, Dr. Felden would have us, each as residents, meet with him one-on-one. And I remember him saying, so how do you envision your career one of these days? And I remember I was still very interested in research. So I remember telling him, I think I still want to do research as a basic science research as part of my career. He says, Matilda, you need to do a fellowship, and then you apply for a KO8. And that was actually the first time I had ever heard about a KO8. And so, so when I went, I ended up matching at the Proctor Foundation at UCSF. And that's where I did my cornea fellowship. And about halfway through my fellowship, I started to apply for my KO8 award. And when I was trying to decide on what topic to study, I was really interested. These were 4 patients that I happened to see during my internship year. So these were examples of patients who came in with various eye injuries, a thermal injury, fungal infection, bacterial infection, acanthamoeba infection. And so it really interested me to maybe pursue looking at corneal opacification or corneal scarring as a research topic. And so corneal disease is actually one of the leading causes of global blindness. And this was a paper that just came out recently in ophthalmology. This was in the study, they looked at the global trends in blindness and vision impairment due to corneal opacity from between 1984 and 2020. And they found that an estimated 5.5 million persons worldwide are bilaterally blind with moderate to severe vision impairment due to corneal opacities, with an additional 6.2 million that are unilaterally blind due to corneal disease. And so clinically, with all of these patients with these corneal issues, you can see neovascularization, tissue destruction, inflammation, and together these different processes caused corneal scarring. And oftentimes, and still to this day, the main treatment for these patients is penetrating keratoplasty. So I became really interested in particular in studying the scarring process. And so what I did in the middle of my fellowship year, I started reaching out to different potential mentors at UCSF to see who would be interested in mentoring me for this KO8 application. And I was really lucky I found Zena Werb. So Zena Werb actually is not, at that point, she had never done any eye research before. So she was a big breast cancer researcher at UCSF. So she had a big lab. She was primarily interested in breast cancer research, also in breast development. But the reason why I ended up joining her lab was because a lot of the processes in corneal wound or in wound healing in general are very similar to basic processes that occur during tumorigenesis. And she was very generous and took me into her lab. And I was nervous at first because I thought, is this the right decision to join a lab that doesn't do any ophthalmology research? And I'd have to kind of figure this out on my own with her mentorship, but it ended up working out really, really well. And looking back, I'm glad I did that because I got to work with a lot of really talented postdocs in her lab. And she was very insightful into guiding me, even though she didn't have the eye background, she really, really guided me in my projects. So, at a molecular level, what happens when you have a corneal injury is you have activation of epithelial cells. And these epithelial cells then in the wound area, it causes the release of cytokines and growth factors. And then that causes the release of immune cells, the neutrophils, macrophages, and then these go on to stimulate growth factors and reactive oxygen species. And ultimately what happens is you activate MMPs, which you heard about a little bit before. And when I started this project, the main roles of MMPs or matrix metalloproteases were that they had really strong roles in stromal remodeling, inflammation, angiogenesis, and cell proliferation. And so one of the reasons why we clinically use doxycycline for patients, when you have those patients with particularly bad corneal ulcers and you're treating them already with all the fortified antibiotics, but they're really thin and you just want to really protect them from perforating, oftentimes we'll treat with oral doxycycline, and it's not so much for the antibiotic aspect of it, it's more for the what the doxycycline does is it blocks MMPs and then it inhibits further matrix cleavage and allows for remodeling. So the matrix metalloproteases or MMPs are a family of proteolytic enzymes that degrade components of the extracellular matrix. So there's a big family of them. They all have this very basic structural, N-terminal basic structural motif. They have a signal peptide that guides them to the endoplasmic reticulum. They start off in an inactive state, and then when they get activated, the propeptide gets cleaved off. They all have the same amino-terminal catalytic domain where zinc binds and then causes activity, and doxycycline inhibits zinc from binding to this catalytic domain. So that's how doxycycline inhibits all MMPs. And how they all differ is particularly in the C-terminal domain. And it's the C-terminal domain that gives the specificity for each of the MMPs to target their individual substrates. So there's 23 human MMPs and 24 mouse MMPs. So initially, MMPs were named by the main matrix component that, for example, interstitial collagenase cleaves collagen. But it turned out over time that people realized that MMPs cleave more than just matrix components. And so that's why the new nomenclature is to actually call the MMPs by its number, because when people were thinking of them by their old nomenclature, they were thinking that the MMPs only had that one specific matrix component target, but it turns out they target multiple substrates. The MMP that I studied in particular was MMP12 or macrophage metalloelastase. So one of the reasons why Zena's one of the things about Zena was that she had actually just discovered during her career, she had discovered many of the MMP and figured out exactly what they did. And she had actually, during her postdoc, she had discovered MMP12. So MMP12 is expressed primarily in the macrophages, and there are different factors that stimulate its expression. And so MMP 12, even though it's called macrophage metalloelastase because it cleaves elastin, it also cleaves other ECM substrates as well. And as we know in the cornea, there is elastic tissue, so it's a good it's expressed in the cornea. But there also are some non-ECM substrates of MMP12, including plasminogen, which is an important factor in angiogenesis. And so at the time I was starting this project, there was a paper that had come out where they had did an in vitro comparison of different MMPs and found that MMP12 was actually the most efficient MMP to produce angiostatin. So it had this very potential anti-angiogenic effect. And also around the time I was starting this project, there was a study that had been published in IOVS. And what they had done was they had cultured these corneal fibroblasts. And at that time, they didn't have very large scale single cell RNA-seq assays. Back then, we were using DNA microarrays. So the group at Hopkins performed this DNA microarray analysis of these corneal fibroblasts that were induced to differentiate. Of the 165 genes in their array, they found MMP12 to be one of the most significantly upregulated genes. So to study MMP12, there in wound healing, I used two different models of corneal injury. So one model was just to perform using an algebra, just to perform an epithelial injury. So this injury just removes the epithelium. And so it's a really good model for looking at epithelial repair. It's doesn't really generate much scarring or angiogenesis. So to look at those processes, I would perform chemical burns using sodium hydroxide. I would soak a piece of filter paper in sodium hydroxide and deliver it to the cornea for that injury. And in that injury, you actually get corneal fibrosis, angiogenesis, scarring. So these are pictures of mice. This is a picture of a mouse cornea right after I did the sodium hydroxide injury. And after the injury, you get this large corneal epithelial defect right away. By 8 days later, it's already completely healed. But then by day 8, you can already, it's hard to see in this picture, but there's already some blood vessels coming in from the limbus, and you've got this diffuse corneal haze. And I just collected the corneas using qPCR. I just measured MMP12 to see if it was, to see how it was expressed. And it was expressed in 2 waves. It was expressed at right away at day 2. It was also expressed at day 6. And that was my expected result because after corneal injury, most of the macrophages come in around day 6. So that was RNA expression. I also wanted to check protein expression. So I did immunohistochemistry and it was following injury, you got upregulation of MMP12 protein expression as well. The other model that I used for my studies was I wanted to look at mice who didn't have MMP12. So these were, this is how these mice were, they weren't generated by me, they had been generated previously. But basically a PGK-NEO was inserted into exon 2 and by northern blot and western blot, it confirmed that the absence of both RNA and protein in these mice. I used flow cytometry in the mice that I had just to and I used qPCR analysis just to confirm that in my mice, I really didn't have MMP12 expression. So, so when I looked at the, so these are images of wild type mice and mice, the MMP12 knockout mice, 3 weeks after the chemical injury. And so smooth muscle actin is a marker for fibrosis. So in the MMP12 knockout mice, they had increased expression of this fibrosis marker. I looked, and I also, so that was protein, but I also looked using qPCR. I looked at, in addition to smooth muscle actin, I also looked at collagen 1 and collagen 3 as markers of fibrosis at 1 week and 3 weeks. And again, in all three fibrosis markers, there was more expression in the MMP12 knockout mice as compared to the wild type mice. I also wanted to see, because one of the substrates for MMP12 was plasminogen, which is involved in angiogenesis, I wanted to see what happens to angiogenesis in the mice. So I performed whole mount analysis and I stained with CD31 to look at the blood vessels coming in from the limbus. And so the MMP12 knockout mice actually had more corneal neovascularization coming in after injury. I also looked at VEGF-A and VEGF-B expression to see if maybe those factors were responsible for this increased neovascularization, and they were upregulated in my knockout mice. And I also did Western blot and confirmed that plasminogen, which is one of the known substrates for MMP12, was differentially expressed in the MMP12 knockout mice. So I also wanted to see what happens because one of the important processes in corneal fibrosis is inflammation. So I wanted to look at inflammation or immune cell dynamics in wild type mice versus the knockout mice. So I crossed my knockout mice with a line of mice where all the immune cells were gloating green, basically. So these were just wild type mice. So you can see all these were presumed macrophages that were very motile in the MMP12, in the mice that had normal amounts of MMP12. But then when I imaged, these were live mice, so mice that were still alive, and I did in vivo confocal imaging, and the dynamics of the immune cells in my knockout mice was severely reduced. So then this led me to look further into the immune cell dynamics using more a whole mount analysis, looking at neutrophils and macrophages following injury. So these are wild type mice and then MMP12 knockout mice, and I use GR1 as my marker for neutrophils. And in the MMP12 knockout mice, 1 day after injury, there was a reduction in the number of neutrophils that came into the MMP12 knockout mice. And so I performed a protein array looking at different chemokines and cytokines that might be different between the wild type and MMP12 knockout mice to try and see if there was to see what might explain this difference in neutrophil infiltration. And it was interesting that CXCL1, which is a chemokine that helps recruit neutrophils, was actually decreased in my MMP12 knockout mice. I also looked at macrophage infiltration. So, so this is, so in the intact mouse, a wild type, and the knockout mice, there are just normal resident macrophages. And then 6 days after injury, so this is at a 10x magnification and a 20x magnification, there were a lot more macrophages that came into the MMP12 knockout mice compared to the wild type mice. And then in the same protein array analysis, it was interesting that CCL2, which is a chemokine that helps recruit macrophages, it was more highly expressed in the MMP12 knockout mice as compared to the wild type mice. So so that was what I had done during my K08 studies. And then at that time, was when I applied for an R01, and I was very fortunate with my R01. I was able to become independent, and I was also very fortunate that I was able to hire a couple of the first people I hired for my lab were Marie Wolf and Pepe Pan, who were very talented scientists. And what we wanted to do is we saw this, we were very intrigued by the macrophage difference, because What happens in fibrosis is you get, not only do you get abnormal inflammation, but you can get prolonged and persistent inflammation, which is what leads to the fibrosis. So we wanted to know whether or not this, this observation that we saw of CCL2 being more highly expressed in the MMP12 knockout mice was really a consequence of, a direct consequence of the MMP12. So the next few experiments we performed was we blocked CCL2 in particular by, we did subconjunctival injections of CCL2 and then performed the injury. And basically we found that by blocking CCL2, you could increase the macrophage recruitment in our MMP12 knockout mice. We also looked at the corneal neovascularization after blocking CCL2 and it reversed. So this is before this is without the anti-CCL2. So in the MMP12 knockout mice, you had more neovascularization, but then when you blocked the CCL2, you were able to block the angiogenesis back to the wild type levels. So basically, to summarize, this is the model that we proposed based on the results of our study. So following injury, you get macrophages that come into the wound. These macrophages then release MMP12. MMP12 binds CCL2. So it's known that a group in Canada found that one of the substrates of MMP12 is CCL2. So we believe that MMP12 then binds CCL2 and activates it. And then this leads to decreased CCL2 binding to its receptor. And ultimately you get, you inhibit angiogenesis, you get decreased monocyte recruitment, and ultimately you get decreased fibrosis. One of the reasons why we thought this result was kind of interesting was because up to this point, most people had kind of considered MMPs as being destructive. You want to inhibit MMPs because you want to inhibit its activity. And so this was actually an example of an MMP where you want its activity because it's actually protective against corneal injury. So that was all in the lab using mice. And so I wanted to see what happens in patients, how MMP12 is expressed in patients. So then with David Huang at UCSF, we collected corneas from patients undergoing PKP or the DULC procedure. So we just collected corneas from these patients so they could be getting their surgery for keratoconus, for corneal scarring. The corneal scarring could be from a variety of conditions, different types of infections or trauma. And we also had control corneas that we obtained from an eye bank. And so what we did was we collected all of these corneas, generated protein from all of the samples at the exact same time, and then ran it through this Luminex assay. And so this was a protein assay where you could expose the protein lysates to all the different MMPs all in the same well. And you can compare different levels of MMPs all at the same time. And so these are the results for the different MMPs that were in the assay. So patients 1 through 6 were the control eye bank samples, and then patients 7 through 27 were the patients who had a corneal transplant. So this is also a summary of what I just showed on the previous slide. So these are all the different MMPs. So there were 4 MMPs in particular where they were significantly upregulated in the patient samples as compared to the control eye bank samples, in particular MMP2, 8, 12, and 13. We looked at MMP12 expression with neovascularization levels. So all the control samples, none of them had neovascularization. Some of our patient samples were the keratoconus patients, so none of them had neovascularization. And then we had other samples that were post-infectious, and those had a lot of neovascularization. And so when we separated into those three groups, again, we found that the MMP expression levels seemed to be correlated with the level of neovascularization. We also looked at scarring. So we broke the samples down into the control eye bank samples. Some corneas that in the exam note report had no scars at all. Some that had mild scars, so some keratoconus patients that had just mild apical scarring, and then other samples that had dense scarring. And so again, depending on the corneal scarring level, you got an increase in the expression levels of the different MMPs. So to summarize this part of the talk, basically the overexpression of several MMPs, in particular MMPs 2, 8, 12, and 13, occurs in corneas from patients requiring keratoplasty. And the expression of these MMPs seems to positively correlate with clinical levels of corneal neovascularization and fibrosis. And so these results just support an important role of MMPs in the corneal repair process. And so for the very last part of my talk, I'm going to talk about, a lot of my research had been focused on corneal fibrosis and scarring. And so clinically, I've been very interested in corneal ulcers because those are the patients where ultimately once the ulcer resolves, they end up with a large corneal scar. And when I went into when I told Dr. Felden that I want to do 50% research, 50% clinical, I thought my 50% clinical was really going to be just my own patients seeing, in my own faculty practice. But a perk has been, an unexpected perk has been that I've been able to work very closely with the UCSF ophthalmology residents at the county hospital there. And so Lawrence Chan is a current third-year ophthalmology resident who's going to be going into retina, and Jackie Lopez is a fourth-year medical student who's going to be joining our ophthalmology residency program. So this, for our study, we looked at patients at the Zuckerberg San Francisco General Hospital. And so it's been known, just for background, it's been known that county hospital patients may have worse presentations and increased hospitalizations. Other ophthalmic studies or other eye studies have shown that there are social risk factors that can influence outcomes. For example, these studies showed that unemployment can be correlated, that there's a relationship between vision impairment and unemployment. Housing conditions can also affect outcomes in ophthalmology. So poor housing conditions have been associated with worse adherence in diabetic retinopathy. It's also been shown that lower household income leads to a higher hospitalization rate for corneal ulcers. And finally, housing problems and drug abuse can lead to ophthalmic hospitalizations. So what we decided to do was, in our specific patient population at the county hospital in San Francisco, we wanted to characterize how these specific social risk factors influence infectious keratitis at our hospital. So we performed a retrospective cohort study. We included all patients who had a positive culture result for either suspected bacterial or fungal keratitis between 2010 and 2021. So we had a total of 174 patients in our analysis. We excluded patients who only had a positive result for viral keratitis. And the outcomes we looked at were loss to follow-up, hospital admission, medication noncompliance, poor visual acuity, and time to corneal ulcer resolution. So 3 risk factors that we looked at were drug use status, housing status, and employment status. And so out of the 174 patients that qualified for our study, there were 69 that were lost to follow-up. And so when we compared, when we looked at those patients, we found that drug use, there was a trend towards drug use and loss to follow-up. We also looked at housing status, so there was also a trend between homelessness and the patients being lost to follow-up. We definitely found a significant effect of employment status. So patients who were unemployed had an odds ratio of 2.59 of being lost to follow-up. We also looked at this lost to follow-up population, looked at medication non-compliance rates. And not surprisingly, the patients who were lost to follow-up also had higher rates of medication non-compliance with an odds ratio of 2.67. For a lot of our corneal ulcer patients, we hospitalize some of them. And so when we look specifically at the patients that ended up being hospitalized for their corneal ulcers, we found, this was also not surprising, but we did confirm that the majority of the patients that we were hospitalizing for corneal ulcers had a larger initial epithelial defect size as compared to the patients who weren't hospitalized. We also found that the hospitalized patients had a worse initial and final visual acuity. And when we looked specifically at the patients who were hospitalized, and we looked at the social risk factors in that population. So in the patients who were hospitalized, about 80% of them had a history of drug use. 69% of them were unemployed, and 57% of them were homeless. So our results, when we analyzed our data, we found that homelessness was associated with medication non-compliance with an odds ratio of 3.48, but so this was a trend. Unemployment also was associated with worse final visual acuity. And we also found that increased age also caused a delay in resolution time of their corneal ulcer. So this analysis basically led us to conclude that there are social risk factors that are important to consider when you see patients with these corneal ulcers. It's important to figure out their employment status, find out if they're unemployed or not. Any history of recreational drug use, their housing situation, if they're homeless or housing insecure, and age can definitely be a factor with rate of healing. My main take-home summary for my whole presentation. So if you take anything away from my talk, basically, there are 3 important points of my talk. So the most important point is probably that in my career, mentorship has been incredibly important. I've been very lucky to have had such amazing mentors, starting way back in medical school, but it's actually, even though I'm faculty, I still have mentors that I still need tons of advice from. So you can never, you never stop needing help from others. You can definitely design a career that fits you. Looking back, I remember when I was interviewing for fellowships, people would ask me, how do you envision your career in 10 years? And I would always say, I envision it a 50-50 split, 50% research, 50% clinical. And I remember people would say, like, that's impossible. You usually have to pick one or the other. But actually, I think I really do split my time about 50-50. So it is possible. So it's possible that you can have a career that encompasses patient care, bench research, and teaching. The teaching was a pleasant surprise for me, and enjoy the journey. I've been truly, part of the journey is working with coworkers. And so I've been lucky to have had the best colleagues. Thank you.
Hi, thank you so much. I had a question in regards to the last study. we're carrying out a similar study here, and one of the things that we've talked about in the past is just like, how do you reliably determine if patients are taking medications? And we've kind of had to just make the assumption that if they're being prescribed medication, that they're adhering to that treatment. But I was curious how you went about it in your study.
Oh, good question. So usually we would ask, some patients are actually very honest about it. They'll say like, I'm honest, I'm not using it. Do you guys use the Epic system for your EMR? So you can tell in the Epic system whether or not they really are compliant, because it tells you whether or not they're really refilling their medications or not. And then also, and I think from also the clinical notes, like patients will say, I'm taking my medications, but then when they bring in the bottles and it's full, like, that they're not necessarily, and, you look at the label and see when it was prescribed and filled, and then you can tell whether or not they're actually using it or not.
Thank you.
Oh, yeah, I have read about tissue inhibitor metalloproteases. So I don't know if you account for that in your study, and if the activity of these inhibitors depends also in the cornea, depends on the metalloproteins that you're working with, like MMP2 or MMP12. I just wonder.
Yeah, so we didn't look, so when we looked at just protein levels, so it doesn't, I didn't show it, but you can actually look at protein activity. So you can do zymograms, and that's how you look at protein activity. Unfortunately, when we did our study, we only got enough from the samples, we only got enough protein to do that one assay. So we decided to just do that Luminex assay just to look at expression. But yeah, the key is to look at activity. And you're right, some MMPs actually, a lot of MMPs actually directly affect other MMPs, and so some MMPs will then cleave other MMPs and then inhibit those downstream MMPs. And there's also a temporal issue also, depending on what time point you're looking at, there's all those dynamics. So we didn't take those factors into consideration, but those are important.
One more question.
Hi. I specifically wanted to ask in relation to the last study, you discussed a lot of factors that were like limiting the ability to follow patients. And I was wondering if there were, and also the ability for patients to like comply with some of the medication regimens. I was wondering if there were any thoughts or directions on ways to support, like on a systemic level, patients in the future, like how that research was going to be used to like find ways to support those patients.
Oh, good question. So right now, we have a large homelessness issue in San Francisco. And if you look in the literature, there really isn't a lot of data on that population. So our data pointed out to us that that's an important issue, that there are issues, that's a particularly vulnerable population to look at. And so we're hoping that we can use our data to get more resources potentially, if we can collect that data and then use that data to potentially get more resources for that particular population, for our homeless population in San Francisco. Yeah. Thank you.
Thank you. So in recognition of being our distinguished visiting professor today, we'd like to present you with this plaque to commemorate your time here. Thank you so much.
Thank you.
Okay, so just some final housekeeping. I hope some of you can join us at the graduation tonight at Casa Larga. It starts at 6 o'clock. We'll have a special toast at 6:45, so try to be there by at least 6:45 if you can make it by that time, and dinner will follow with the program. And there will be no rounds in July or August, correct?
Well, you're trying for a low vision.
There's no rounds in July for sure. We're trying to get something on the books for August.
But we've got to check people's vacations.
Yes, all right. So thank you all it's a great academic year for rounds. I appreciate everybody's attendance. Have a great weekend.